All right, so if you were just to block it out, you've got the pyruvate dehydrogenase complex over here. And what happens is you're going to have pyruvate's going to go in. We get acetyl-CoA out the other side. And we'll also have some CoA go in. We're going to have a carbon dioxide come out. We're also going to have an NADH come out. Okay, so those are the things we have going on in this process. Have I missed anything? I don't think so. And overall, we talked about this last time. I'll run through it again. Well, then we can run through the cofactors specifically. We'll probably gloss over thiamine pyrophosphate because we got it last time. But we've got pyruvate comes into this complex. It interacts with thiamine pyrophosphate, and we end up with two of those carbons from that pyruvate end up on the thiamine pyrophosphate as its hydroxyethyl group. The other is the carbon dioxide leaves, right? So this carbon has been extra oxidized. You think about it as this bond right here was bound to a carbon, now it's bound to, now we're bound to oxygen. So that's an oxidation step. So there's some energy here. That energy, when this can then be oxidized to make the acetyl group, and that's going to reduce this lipolysine that picks it up. The lipolysine will then hand off that acetyl group to a CoA, and then the acetyl-CoA is going to take that off to the citric acid cycle. We now have a reduced lipolysine here, or lipoate. So we have this reduced lipolysine here. That lipolysine now needs to be reoxidized so we can do this again. And so doing that, it's going to hand off its electrons along with these two hydrogens to FAD to make FADH2. So this has now been reduced. But this is bound to the enzyme. It can't float off to the electron transport chain on its own. FAD doesn't do that. So our FADH2 is going to need to be reoxidized. And so it's going to hand off its electrons plus hydrogens to one NAD takes one hydrogen. The other one goes out as H+, but we end up with two electrons going in here. These are all two electron processes. In all these oxidation reduction steps we talk about, this term are two electron processes. It doesn't mean that one electron processes don't happen. They just don't happen in the stuff we're going to talk about. Yes?
So are all the blue lines attached to the lysine part of the same molecule, or is it at the same molecule in different steps, or one big molecule?
So this is a lipoate, and we'll look at it kind of drawn differently.
Okay.
When we go and look at lipoate specifically. But this is the business end of the lipoate, okay? And then the way it gets bound to the enzyme is through the end of the lysine. So you can think about, you've got lipoate, it's got kind of a tail to it, and then it gets an even longer tail or arm to swing around with you add a lysine, because you've got lysine with those 4 carbons and the nitrogen.
Are they 3 separate lipoates? Or is
no,
it's
all the same. That's a great question. Good question. So this is all the same thing right here. Same molecule going through. Yes?
Is, this is just a general question, but is FAD always, attached to something?
Yeah.
Is what?
Is FAD always attached to something?
FAD, yes. Yes, it's always going to be in, it has to be made and get into something to begin with, but it's always bound by an enzyme. And how much energy, when it's reduced, it actually kind of carries depends on its environment. We'll talk about, we'll maybe have time to get to talk about phosphate to ATP ratios, or oxygen to ATP ratios, oxygen to phosphate ratios at the end. You'll see why I missaid it that way. But this is in this case, this is worth about 2.5 ATP. It's holding about 2.5 ATPs worth of energy, at least, because this is 2.5 ATPs worth of energy when we get to electron transport chain. Usually it's going to be 1.5 in most other cases where I see it. Other questions? All right, so we talked about thiamine pyrophosphate, so I'll just go over it pretty quickly here, and then we'll move on to talking about lipoate. So here we have our thiamine pyrophosphate. You can see it. This is in our unloaded form. This is our loaded form. So remember, for all of these, you need to be able to recognize a name, and then you're going to have to tell whether it's oxidized, reduced, or loaded, unloaded. Depending on the molecule, it may have one or the other, okay? So this doesn't have an oxidized reduced. This has a unloaded, right? It doesn't have the hydroxyethyl group on it, and it has the loaded form. And you need to be able to draw the other form. All right, so you're not responsible for this whole thing here. This would be given to you. You need to be able to recognize, oh, yeah, that's thiamine pyrophosphate. And then you need to recognize, is it in its loaded state or unloaded state, and be able to draw the other one. Questions? Okay. So let's go on and then look at lipoate. We'll have a little more detail down here. So when we talk about lipoate or lipolysine, I kind of use those interchangeably. But here, this figure is nice, right, in that it shows the lysine. And I'll accept either of those, if you want to call it lipoate or lipolysine, regardless of what's drawn. So here what we have is this is the actual lipoate. Okay, they're saying lipoic acid. Really, it's neither because it's attached through an amide. And you'll see things will often get attached to enzymes through an amide at the end of a lysine. And then here's the lysine part, and then this is the chain, the backbone in the actual protein. So this is what our oxidized form looks like. We're kind of familiar with this, right? That we have a disulfide, we can make our oxidized when they're linked together. That's our oxidized form. Our reduced form is we add some hydrogens, right? Just like in organic chemistry, when you have more hydrogens, something is more reduced. And then we have our acetylated form over here. That's the form that actually has the 2 carbon on there. So you don't need to remember acetylated. I would call this loaded. So this one actually has all of those things, right? We can talk about it's loaded, it's oxidized, or it's reduced, depending on what you see. Yes?
So this is, can you show like in a diagram which
Back on the previous one? Yeah.
it's looking like it's like reduced and then oxidized. This is like
So here it's oxidized.
uh-huh.
Here it's reduced. They actually have them labeled. And then here, I would call this loaded.
So it's just like kind of like a carrier, like just like carrier that's delivered.
Right, we've got a lot of carriers here. It's a carrier, but it's also in going from here, right? This is, when we think about our 2 carbons here, this is more reduced than this, right? So going from here to here, we have an oxidation event happening. If something is oxidized, something needs to be reduced. So this is getting reduced, even though we're going to call it its loaded form. And then once it's gone there, now we can call it its forward or reduced form. So it's a carrier, but it's also captured some energy here that's going to be transferred here, that's going to then be transferred here, that's going to get us 2.5 ATP.
Okay.
So that's important as well. Okay. Questions on lipolysine?
And I'm sorry, one more question. What's the group that's attached to it? Like when it's loaded?
It's an acetyl group.
Okay.
This is an acetyl group.
I know you crossed out acetylated, but if we wrote acetylated, would you mark it wrong?
That'd be fine. Okay, so then after we have that acetyl group is then going to get transferred over to a CoA. So this is called coenzyme A is the full name as written on the page here. Off on the right, we see something we've seen before. What is it?
Uh,
ADP, right? We've got ADP on another one of these cofactors that makes a good, remember, it's a good handle. It's something that there are lots of enzyme pockets that have evolved to bind this. So good handle. That's really what it's doing there. Okay. Don't worry about the penthoic acid in the middle. Okay. And then the business end of this is this thiol group. Right. So what we're going to do, you need to be able to recognize, hey, that's the same sort of thing as before. You need to be able to recognize all that. But usually the way we'll draw it is we will really just draw it like this. We'll write CoA, we'll put an S on the end, and it'll be SH or it'll be S-acetyl group. Does that make sense? Okay. So separate from that, because now that acetyl group is going to run off to the citric acid cycle. Now we have to reoxidize. All right, let's go back up here. We have to reoxidize this lipoate so that we can do another cycle. And so that happens by it interacting with FAD. And we've seen FAD before. Right? And so what we have here is, once again, I keep using these same figures. I'm going to go ahead and, oh, it doesn't like black. Okay. I'm going to cross this one out. That's a 1 electron transfer. We're just doing 2 electron transfers in this class. Okay. So we're going to have, we start, this is our oxidized form right here. Okay. And then this is our reduced form. This one says it's reduced, already oxidized there. And so this, we got no loaded, unloaded here. But this is oxidized. And you see where the hydrogens went on? Okay, and then if you wanted to convince yourself, what I like to do, I think I've done this for you guys before, I like to bring in H dots. I can bring in an H dot. So let's say we've got, let's just look at this top ring. So this is our oxidized form. So you don't have to show this mechanism. This is just a way to think about the oxidized versus the reduced. So I've got that. And so if I think about bringing in an H dot, I can do a little 1 electron arrow there to there, there to there, there to there, right? And then I can bring in another H dot over here and bring this up to make a bond there, and that one to make a bond there. Does that make sense? Do you see how we now brought in 2 electrons? So you guys see hydrogens, you think about atoms and stuff like that, but these are all, it's all about the electrons, right? Hydrogens might come with them or not. Yes?
Isn't it like a radical? Yeah.
Yeah, but we're doing a 1 electron radical arrow thing there. I'm doing that to make that. But I don't need you to do that.
Yeah.
I did it as much to say, hey, this is how you can think about going from here to there, and think about that I'm adding 2 electrons. Does that help?
Yeah, I was just like, cause like anytime you're radical, it's like unstable, like thought.
Well, you can have radical reactions that happen regularly in enzymes and things like that. So.
Hydrogens are just kind of the best explanation.
And there are stable radicals as well, but usually a radical is reactive. That's the right feeling. If you ever see a radical, you should think unstable, dangerous, whatever, you know. All right, and then once again, we have our NADH that we need to recognize. We're not concerned with what the stereochemistry is like this figure has, but otherwise it's a great figure. And so we'll have 2 electrons plus 2 hydrogens, but really we've got 2 electrons that are going in and we end up with adding a hydride to this from here. Okay. If you wanted to push your arrows, you add your hydride, you push this down, you push that up onto there. Okay. Any questions? Okay, and this is the final acceptor that runs off to the electron transport chain. Okay, this is what can give us, go to the electron transport chain and produce a dump off 2 electrons at the very beginning of the electron transport chain and get us our full 2.5 ATP. Okay, do you want me to go over this again?
Yes.
Okay, so once again, doing the cofactors, once again, we have our pyruvate. It comes in, is picked up by the thymine pyrophosphate as a hydroxyethyl group, and we're losing a carbon there. This carbon got oxidized, so this is a little more reduced, right? Okay, and then I'll stop there for a second. Okay, and then so this hydroxyethyl group is then going to get handed off to a lipoate, which is attached to the lysine. And in the process of that, we're going to reduce the lipoate and oxidize the hydroxyethyl group to make it an acetyl group. Right. The acetyl group gets handed off to acetyl-CoA, and that goes off to the citric acid cycle.
How is this like favored evolutionarily? It seems so overly complex.
So it is complex, but especially how it's favored is everything's being handed off.
Mm-hmm.
So it's more efficient in that we don't make a hydroxyethyl group that's floating around solution, right? And then it has to diffuse back into the next enzyme. So we often have, in many cases, not all cases, right? So for glycolysis, it doesn't happen. For the citric acid cycle, I think it's a little debated whether you have things in membranes, but we get to things like the electron transport chain, and you have something handed off to something handed off to something, and it's really efficient and happens fast because you don't have to have your reagent, your products from one reaction diffused to become the reactants for the next one.
So it's an assembly line, basically.
Yeah, it's called substrate channeling, and I'll name it more once we get to a different example of it. But this is substrate channeling. Another thing that I think is kind of fun is, well, I think a lot about, I watch a bunch of cosmology videos, so things going out in the universe, is there life in the universe, all that sort of stuff. And what's really interesting, I actually listen to some podcasts on it too, so you think about it, there's just nothing in the universe more complex than life. It just isn't. You think about all the sorts of things and all the levels of complexity that we never even get to in this class. So, in the research group I'm working in, I'm reading some stuff. There are parts of the nucleus that are, this part's active and this part's not active, but they're not on the same histones, and they somehow get together and they hang out in this area. So they have domains from different histones that have kind of loosened up, or I shouldn't say different histones. You've got different nucleosomes from different parts that come together in this region, and this region is active, but these nucleosomes that aren't next to each other aren't inactive. It's amazingly, dizzyingly complex, but it works.
Are
So
the things sticking out of the membranes, like the acetyl group and the SHs, like not the membranes, but like
the protein.
Yeah, is that actually sticking out, or they just fall?
This is just a cartoon representing it, and I can't. It's probably not sticking out.
Okay.
It's probably happening inside, or at least on the surface, kind of buried in a cleft, but I don't really know what this looks like. I mean, we do know what it looks like, but it doesn't really help, right?
Yeah.
We know it looks like, well, not that. Let's go back to lecture. It is in the mitochondria. I don't know that if it's in the membrane, or I don't know that it's in the membrane. I actually don't know where it is. Do you know from either reading your book?
I thought I saw something, but I think I forgot.
Okay, it is in the mitochondria. I don't know if it's in the membrane or not. But that's what it looks like. Yeah. doesn't help me in terms of your question, though. Okay, so we're back here. All right, so that's off to the citric acid cycle, right? And so now we need to repeat, we need to regenerate our enzyme complex. We have this reduced lipolysine, which, it's work to reduce it, but there's energy there. There's ATP to be had. And so we're going to have our lipoate that's reduced is going to hand off its electrons to FAD to make FADH2, which is now reduced, which is always enzyme bound. So it's going to have to hand off its electrons to something else. In this case, that something else is an NAD, makes it NADH, and that is off to the electron transport chain. Okay, questions on that? So you should be able to do the things we talked about for those 5 cofactors. You also should be able to walk through this description without a diagram. Okay. It's like a sentence for each. Talk about oxidations and reductions.
But you said we don't need to know the enzymes.
You don't need to know E1, E2, E3. I don't.
Does that include?
Oh yeah, that's you don't need to worry about that. Okay. All right, so we're going to move on and talk about the citric acid cycle. Here's the whole citric acid cycle. It's a cycle. Okay? But I really do mean it. It's a cycle. Okay? And how I mean it's a cycle, let's talk about that. Okay? We can't see anything just yet, right? I'm not going to follow it around. Let's do it. It's a cycle first. So we've got this thing. Let's call it A. Okay? Just for ease here. A becomes B, right? Becomes C, becomes D, becomes E, becomes F, becomes G, becomes H, becomes A. Right? Okay, so the one thing that I do want to emphasize here is, so we've got these things. So A becomes B, right? B becomes C, C becomes D, D becomes E, E becomes F, F becomes G. I do have a point here. G becomes H.
Rewriting the alphabet.
I'd have you sing. H becomes I. Okay? So we have those, and then I becomes A again, right? Oh no, we didn't have an H. We don't have an I. We have H becomes A. So what happens, these are the main intermediates, right? They're the things right here. So what happens if I go and I add these all up? Well, B's cancel, C's cancel, D's cancel, E's cancels, F's cancels, G's cancels, H's cancels, and the A's cancel. What do I get? Nothing. Okay? So all of these things in the actual intermediates, you just go around and around and around, and you're not doing anything. So the important part of the cycle is the business end. What you're doing is what comes in and what comes out. So it's these arrows on the outsides. All right, and I need to get you printed out some of these. This is the figure from your book. But let's go ahead and I'm going to go ahead and magnify this out. And let's talk about some of those things. So what really happens in the citric acid cycle? Well, it does important stuff, yes. What we need to know about it, well, you do need to know all the structures and the enzymes and same stuff we needed to know for glycolysis. Okay, but what happens is, let's take this part right here. We get this part, we get acetyl-CoA, and that's going to go and give us CoA, right? In terms of that arrow. We're just picking off the things on the sides. Here we have an NAD plus becoming NADH, and we have a carbon dioxide come out. So NAD plus becomes NADH plus a CO2. And there'll be another proton in there, but we don't need to, we're not going to count protons, okay? I mean, they count, but we're not going to add up protons. All right, so then we're down here. We have an NADH, right? And that NAD plus becomes NADH, and we have another CO2 come out. Plus we have a CoA coming in.
Where's the SH?
That's the SH on the end of the acetyl-CoA. So if we go back up here real quick, it's that red SH right there. Okay, it's really the business end. It's what's doing the work. CoA, the rest of CoA isn't kind of some magical thing outside of it's just a big handle. Okay, so here we've got NAD plus. I'm going to run out of room here. We're going to get another NAD plus. We got an NADH, right? We got another CO2, and I'll write it down here. And we ended up, well, actually, we're adding a co-SH, right? A CoA. So we're going to add another CoA here. All right. Now, so that was right here. Let's go here. We've got a GDP plus phosphate to make a GTP, and there our CoA comes back out. I already forgot. It's a GDP plus a phosphate goes to make a GTP plus a CoA comes out, right? Okay, next step. Anything coming out? Oh, this Q thing. We'll have to talk about the Q thing in a bit. That's a ubiquinone, and it comes out as a ubiquinol. And we'll look at the structure of that in a minute. So it ends up the ubiquinol is reduced. All right, let's go back down here. Let's see what else we got. I think we're almost done in terms of things coming in. We get a water, right, going in, and then we also have another NAD, NADH. Let's go ahead and add those to our list. Okay, I've got H2 over here, and then we have an NAD plus goes to make NADH. All right, so what's our overall reaction here? What things can we cancel out? This is going to tell us what actual work happens with the citric acid cycle. So I can cancel out, I've got a CoA, right? And I've got a CoA. Did I do that again? I don't. I've got acetyl-CoA and then a CoA comes out. Okay, great. I have the GTPs, I have the water. It looks like that's all I can cancel. That's disappointing. Okay, but we can write the rest of this out, right? We can certainly sum some things up though, right? How many NADH do we have? 3. How many NAD plus do we have? Okay, how many CO2 do we have? 2. Okay, so why don't we go and write this whole thing out then? We've got acetyl-CoA goes in. We have 3 NAD plus go in. We have a GDP plus a phosphate goes in. We have a ubiquinone goes in. We have a water that goes in, right? Okay. Did I miss anything going in? I don't think so. All right. And then what comes out? Well, we have 3 NADH, we said. We had 2 carbon dioxide that come out. we have a CoA that comes out. We have a ubiquinol that comes out. And we have a GTP that comes out. Did I miss anything? I don't think so. Okay, so that's it. That is the overall reaction for this citric acid cycle. It's all the things that come in and out on the sides. All right. The point here is, so we have, well, let's talk about a little bit of this. So we've got a bunch of energy molecules here. We'll get back to them in a second. Where do these two carbon dioxides come from?
We're going down the number of carbons in our chain. Like they're deep, like they get cut off, right?
They get cut off, but where in this overall reaction, where are they coming from? Their products, where are they on the reactant side?
This CoA.
What was that?
The CoA.
Not just the CoA. The CoA comes out, so that's not coming from the CoA. It may be, no, not the quinone thing. The acetyl group.
Oh.
Right? Which is maybe what you meant, Sophia. But the acetyl group has two carbons. That's those two carbons. Okay? For those of you taking the MCAT, it's not actually, I don't want to overcomplicate it. But we're going to say it's those two carbons, okay? But what it is, it's not actually those two carbons the first round. If you look at the mechanism, the two carbons that come out are a different two carbons than those two carbons, but we're just going to pretend they are, okay? So it's slightly more complicated if you're studying for something else or somebody else really cares. I don't care that much, but you do have to go around the cycle more than once to get those two carbons out. So we get those two carbons, and then we get a bunch of energy molecules, right? We have this water that goes in. I don't even care much about that water, especially because I'm too lazy to actually keep track of my hydrogens, right? I don't care. We're not going to worry about the hydrogens here. Questions? All right. And so when we look at the products here, we have NADH comes off, and we have ubiquinol comes off, and these are both destined. These both go to the electron transport chain. Or these four go to the electron transport chain. The GTP is worth energy, but it can be interchanged with ATP for no energy. So you can take all the whatever TPs, NTPs, and you can make them all into the other NTPs at no energy cost. They're all about the same energy. So it'll just get used directly somewhere or turned into ATP and get used. Questions? All right, let's talk about this ubiquinol then. What do we have for a figure here? Oops. We're going to go down this little bit. All right, right here we have this ubiquinol. Depending on what citric acid cycle you've learned before, you might have learned that FADH2 is made here. And in fact, it is. And so depending on your figure, but FADH2 never leaves the enzyme, right? So it can't go to the electron transport chain. So what's going on here, if we want more detail, is that this is FAD. It's FAD in the enzyme, in the dehydrogenase, going to make FADH2. And then we need to regenerate that, just like we saw before in the pyruvate dehydrogenase complex. And the way that happens, the way we reoxidize the FADH2 is we're going to take ubiquinol and make ubiquinone. I'm sorry, we're going to take ubiquinone and make ubiquinol. Okay? And then this can go to the electron transport chain. This is never floating around.
How is that spelled?
Ubiquinone, ubiquinol. Well, let's talk about that. So we have to learn a new cofactor here. We're also going to see this in photosynthesis. It's not the exact same one, but it's pretty close. So this right here is ubiquinol. Let me pull it over the side and we'll do an oxidized reduced. Okay. If we want, so type it. You probably have, in terms of having seen it before. And then we'll have, that's the reduced form. And then we have ubiquinone. And ubiquinone is going to look like the same molecule, except we're going to have, it's a quinone, essentially. We have these double bond O's instead.
That's a test.
Oh, whoops. Sorry.
Yeah.
Did I have 5 bonds to carbon? That's embarrassing.
I think I don't have my organic one final,
So ubiquinone, what we've done is we've oxidized this, right? And so, well, that's the oxidized form. You reduce it, you add a couple hydrogens, right? You can see we have 2 more electrons in there. So it's a 2 electron carrier. It's going to be worth 1.5 ATP when we get to the other end. Questions?
What's the 10?
The 10 means that we have this thing happen 10 times. So it's like a lipid tail. And it actually sticks in the membrane in the electron transport chain. And it sticks, so we get this, I think we'll get this off of the membrane in terms of the transfer as well. So this becomes reduced, it wanders over the electron transport chain in the membrane, it dumps off its electrons, it becomes oxidized, it wanders back to the dehydrogenase complex as well. All right, so we got 2 minutes left. Let's just look at the citric acid cycle and talk about some general characteristics here. So you can't see it all that well from where you are, but you probably can see some things. What would be good sites of regulation here? Right.
Isocitrate to like heal fluoride.
Right. So right here, okay, where we go from oxaloacetate to citrate, right here and right here. Those are great places to regulate this. Okay. This is not a case where you take the citric acid cycle and you run it backwards sometimes and forwards sometimes. At least do it from a controlled perspective. But you might, the citric acid cycle might be running faster or slower depending on the needs of the cell. All right, so good places of regulation are here, here, and here. But really the only places of regulation in terms of making it go forward faster or slower. That said, the citric acid cycle can be regulated kind of on a longer term based on how much of these enzymes you have, right? And also by how much of each of these reagents you have. So this is an important point about it's a cycle. It's a cycle if we do a round of a citric acid cycle, let's add up how many A's, B's, C's, D's, E's, F's, and G's, and H's we have. Add up all the concentrations of all of them. Okay, we're not telling you what the concentrations are. But let's say it's 100. Okay, I run one time around the citric acid cycle. What's my concentration? I added 2 carbons, right? How many carbons came out? 2. So what's my concentration of all those intermediates after? It started with 100. How many did I get at the end? 100, right. What if I add 1,000 acetyl-CoAs to it? I run 1,000 cycles. I run 1,000 cycles of it, right? How many? What? I have concentrations of all my intermediates up. What have I got?
1,000.
No, 100. It doesn't change because I add 2 carbons and I get 2 carbons out. I cannot increase, this is part of it's a cycle. I cannot make more of any of these things by adding acetyl-CoA, or adding the acetyl groups. If I want to make more of these so I can run more citric acid cycle, which happens in cancer, okay, I need to add it from, I need to deliberately add more of the intermediates, okay? That's an important point. All right, sorry, a minute over. You guys have a great weekend. I hope to see you on Monday. And I hope you